Hello everyone and welcome to my video lecture. Okay, so our topic for today is meiosis. Okay, so all those who have confusion in meiosis, this is going to solve all your problems. Okay, so let's start with meiosis. I have already made a video on mitosis and I have uploaded it. Okay, I have also made a video on cell cycle. So the link will be shown up on your window. So make sure that you watch the mitosis video before you come here, because if you understand mitosis, then meiosis is 50% same, or I will say maybe more. Okay, so let's start with the video. Okay, so what is meiosis? Meiosis is also called as reductional division. If you remember, mitosis is called as equational division because the number of chromosomes in the daughter cells, they remain same. But meiosis is called as reductional division because the number of chromosomes in the daughter cell, it decreases. So how does that happen is, so this is just an overview of meiosis. Suppose if this is a cell, correct, and if suppose this cell has 4 chromosomes, so the chromosome number of this cell is 4. So now when this cell undergoes meiosis, what happens is meiosis is divided into two parts. There is something called as Meiosis-1 and there is something called as Meiosis-2. So what is going to happen is, when this cell undergoes Meiosis-1, okay, there will be two daughter cells formed, correct? That is just like mitosis. However, the chromosome number in each of these daughter cells will be half. So this cell will have only 2 chromosomes, this cell will have only 2 chromosomes. Okay, and this is called as Meiosis-1. Okay, Meiosis-1 is made up of all the same steps like mitosis, prophase-1, metaphase-1, anaphase, just that in front of the steps you add 1. So it is prophase, metaphase, anaphase, telophase, cytokinesis, and these two cells are formed. Then what happens is, each of these cells further undergoes meiosis 2. So if I want to draw those cells, then okay, so how many cells will be produced? There will be 4 cells which will be produced in total. Now the chromosome number is not going to change. Okay, please remember, now from 2 the chromosome number will not become 1. Each of these cells will have 2 chromosomes each. Okay, and this division is called as meiosis 2. Okay, so obviously it will consist of prophase 2, metaphase 2, anaphase 2, telophase 2 and cytokinesis 2. So please remember, the only difference over here is this entire part of meiosis 1. Meiosis 1 is reducing the chromosome number to half, and that is why meiosis is called as reductional division. However, if you see meiosis 2, then meiosis 2 is same as mitosis because the single cell is dividing into 2 daughter cells. Chromosome number is also remaining same, so meiosis 2 is same as mitosis. Is this clear? So we are going to study this entire meiosis today. Okay, we are going to see how this chromosome number in meiosis 1 decreases to half. Okay, so let us start with meiosis and let us start with meiosis 1. All right. Okay, so first of all, we start with meiosis 1. Okay, so now meiosis 1 starts with prophase 1. All right, so let us first draw the first stage, which is your prophase 1. So this is the cell, correct? Now what happens in prophase? All the same things that happen in the prophase of mitosis. So first thing that happens is the nuclear membrane begins to disappear. So I am drawing a dotted nuclear membrane. That means that the nuclear membrane or the nuclear envelope, correct, the nucleus has a boundary which is called nuclear membrane. It begins to disappear, correct. Also please remember, before meiosis 1 starts or before prophase 1 starts, obviously the interphase is present. So the part of the cell cycle which is the interphase, that is preceding meiosis, it precedes mitosis, it is also present before meiosis. So please remember that. Is this clear? Okay, so now, so this is your nuclear membrane, right? It is disappearing. What will happen inside the nuclear membrane? The chromatin is present. You remember interphase, chromatin is present in interphase. It is going to condense into chromosomes, correct? So that same thing is going to happen, but please remember, in the prophase 1, so let me write down the name of this stage. In the prophase 1, there is a slight change in how the chromosomes are arranged. Okay, in prophase 1, similar shaped chromosomes, they attach with each other. So if I say that the chromosome number of this cell, suppose it is 4, then what is going to happen is the chromosomes are going to be present attached to each other in pairs like this. Can you see this two pairs which are formed? I will do one thing, I will draw a bigger diagram so that you can easily see. So I am drawing a larger nucleus over here, okay, and now inside I am drawing the chromosomes. So this is one pair of chromosomes, they are both attached to each other, and this is the second pair of chromosomes. Can you see? So this arrangement occurs in prophase 1. So by the time prophase 1 ends, you have these pairs of homologous chromosomes. Okay, I am writing the word over here. What is the meaning of homologous chromosomes? So chromosomes which are similar in shape and size. Okay, for example, if I draw 4 chromosomes over here, so these first two are similar in shape and size, so these form a pair of homologous chromosomes. These two are similar in shape and size, they are homologous chromosomes. If I consider this one and this one, they are not homologous. So what happens is these chromosomes which are homologous, they pair up. Okay, so their chromatids attached. Okay, and similarly these two will pair up. So when is this happening? This is happening during prophase 1. Now whenever we study meiosis 1 and we study prophase 1, so prophase 1 is further divided into 5 steps. Okay, in this video I am not going to go into that detail because then it will be confusing for people who don't know about meiosis. Okay, so if you need that, I will make a separate video on the stages of prophase 1. See, there is leptotene, zygotene, pachytene, diploid, dikinesis, all those stages are there. So prophase 1 is further divided into 5 steps, but I am just going to show you what is happening in prophase 1. I am not going to teach those stages because today I want to make this process of meiosis clear. If this is clear, then you can understand, or then prophase 1 becomes easier to understand. Is this clear? Okay, so let us resume back to the lecture. So what happens is this similar grouped chromosomes, these similar shaped chromosomes, they group together. So by the end of prophase 1, you have these pairs. If the chromosome number is 6, then you will have 3 pairs. Okay, only chromosomes which are similar in shape and size, they attach with each other. Please remember this. So if the chromosome number is 4, these two groups will be formed. Such groups are not formed in prophase of mitosis. Please remember, if this was prophase of mitosis, then the diagram would have been like this. This would have been the cell, correct? This would have been the nuclear membrane, and you would have got 4 separate chromosomes. Please remember this. This grouping doesn't occur in prophase of mitosis. Okay, and because of this type of grouping of homologous chromosomes, you get the reduction in chromosome number. We will see how. So anyway, prophase 1, coming back, what else happens in prophase? Do you remember? The centrosomes begin to move away from each other. So the two centrosomes, they are moving away. Obviously they will be having the centrioles in the case of animal cell. If it is a plant cell, centrioles are absent, and the spindle fibers, they begin to form in the G2 phase, you know that. So everything else is the same in prophase 1 of meiosis. The only difference is homologous chromosomes group together, okay, and these groups are formed. Now please remember, these groups of chromosomes, suppose if I draw this single group of chromosome, then these two chromosomes are also called as bivalent, or they are also called as tetrad. Tetrad is 4. These two chromosomes have 4 chromatids between them, right? Because one chromosome has 2 chromatids, so that is called tetrad. Bivalents mean 2 chromosomes. Please remember this word. So homologous chromosomes are also called as bivalent or tetrads. Is this clear? Okay, so that completes our prophase 1. So all the steps are same as the prophase of mitosis. The only difference is homologous chromosomes are formed. Now we go to metaphase 1. All right, so what happens in metaphase 1? So I'm going to rub this off. So in metaphase 1, again the arrangement is going to be very similar. So by the time prophase 1 ends, what is going to happen is this membrane which is there, that entire membrane will disappear. So your nuclear membrane has completely disappeared, by the end of prophase 1. Now coming back to your next stage, which is metaphase 1. So this is the cell, right? Now both the centrosome have moved at the opposite poles, correct? They have the centrioles inside them. What about the chromosomes? The chromosomes again arrange themselves on this central metaphase axis. So how will they be arranged? So now you have two chromosomes which are attached to each other. This one single pair of homologous chromosomes, this is the second pair, correct? So this is how the arrangement occurs. Please remember, the arrangement is not like this as in metaphase. Why? Sorry, as in mitosis. Why? Because now you don't have all chromosomes which are separate. The chromosomes are actually linked to each other. That is why the arrangement is like this. So that is why this is very, very important to remember that tetrads or bivalents are formed. So do you understand this arrangement? Now, what else will happen? You have to show the spindle fibers or the microtubules. So the spindle fibers will go and attach to the centromeres of the respective chromosomes, okay? And if you remember, to the centromere are attached the proteins called as kinetochores. Kinetochores act like a glue or an adhesive, and they help in attachment of spindle fibers or microtubules to centromere. Is this clear? So this is how the arrangement is in prophase 1 and metaphase 1 of meiosis 1 stage. Now rest of all the steps are same as the ones that we studied in mitosis. So what is going to happen next? Next is, so metaphase 1. Next step is anaphase 1. So what are we going to show? Obviously the cell is going to elongate slightly, it will become elliptical. Why is that going to happen? That is going to happen because the two centrosomes are pulling the spindle fibers towards them, and that is why the shape becomes elliptical. So centrosome number 1, centrosome number 2, spindle fibers. Now what is going to happen? See, in mitosis what happened was a single chromosome was pulled towards both the ends, and that is why the sister chromatids of the chromosome they separated. What are sister chromatids? So if this is your single chromosome, then these two chromatids attached to the same centromere are called as sister chromatids. We have already done this, correct? So in mitosis sister chromatids separated, but now what is going to happen? Now this chromosome is going to be pulled upwards, this one is going to be pulled downwards. Similarly the same is going to happen for these two. So now sister chromatids will not separate, entire chromosomes will separate, and because of that now we are going to show one chromosome over here, one over here, the other over here, and the other one over here. Is this clear? And if you realize, now suddenly what is going to happen is this is going to form one cell, this is going to form other cell, and the chromosome in each cell, the chromosome number in each cell has already reduced. Okay, so is this clear? This stage is called as anaphase 1. Please remember to write down that one because we are studying meiosis 1 right now. Okay, next stage will be what? Next stage will be very, very easy. It will be telophase 1. So in telophase 1 what is going to happen? So again the cell, correct, then both the chromosomes have reached towards the respective poles. Then we have to show the centrosome as well. So this is the centrosome, another centrosome. Now what happens is, see, in telophase 1, please remember, normally what happens in mitosis, so these chromosomes, they decondense back into chromatin, correct, or they unfold into chromatin, and the nuclear membrane is reformed. That is what happens in the telophase of mitosis. Now in meiosis, most of the cells will follow the same thing. That means what is going to happen? These chromosomes will decondense into chromatin and nuclear membrane will be formed. However, in some organisms, the nuclear membrane is not formed and these chromosomes also do not decondense. They remain as they are. Why? Because after meiosis 1, meiosis 2 is going to start. And in meiosis 2, prophase 2 is going to be the first stage. So anyway in prophase 2, you will have to remove the nuclear membrane, and if chromatin is present, you will have to condense it into chromosome. So to prevent that unnecessary wastage of energy, these chromosomes remain as they are in telophase 1, okay, and the nuclear membrane is also not formed, but that is in some organisms. So right now I am going to show you how it happens in most of the organisms. So we follow the same rule of mitosis. So I am going to show the nuclear membrane, okay. So the nuclear membrane I am showing, which is reformed, correct, and next is the chromosomes decondense or uncoil into chromatin. So this is the chromatin which is formed. Is this clear? So if you want to avoid confusion, you remember the same thing which happens in mitosis. However, some cells in meiosis, they keep the chromosomes as they are and they do not form the nuclear membrane. Okay. So this is your step which is called as telophase 1. Now last step of meiosis 1 is cytokinesis, correct. So what happens in cytokinesis? So you know that a cleavage or furrow is formed. Now the karyokinesis is complete. What is karyokinesis? Let me write down that word in case any of you has forgotten. Karyokinesis is nucleus splitting up into two parts. So originally you had a single nucleus, now it has split up into two nuclei, correct. So that is karyokinesis. Karyon is a nucleus. Now that is finished, that is over. Now you need to split the cytoplasm. So now we are going to start with cytokinesis, which is the last step. So this is called as cytokinesis 1. What is going to happen? The furrow is going to be formed, okay. So the cell will assume this shape, okay. This is called as a furrow or an invagination. Then again we are going to show the two centrosomes. Then we are going to show the nuclear membrane, and we are going to show the chromatin inside the nuclear membrane. Is this clear? And then what will be the next step? The furrow will still increase in length, and what is going to happen is you will have two cells. So you have cell number 1 over here and you have cell number 2 over here. Is this clear? So that ends your meiosis 1. This is cytokinesis 1. You have two cells. However, in each cell the chromosome number has reduced. Why? Because if you see over here, these two chromosomes will enter this cell and these two chromosomes will enter this cell. So the original parent cell had 4 chromosomes. Now each of the daughter cells has only 2 chromosomes, and this is called as reductional division. So if you understand this, then your meiosis will be clear, because meiosis 2 is same as mitosis. Is this clear? Okay, so please remember, in meiosis 1, homologous chromosomes are attaching to each other and they are forming a structure called as tetrad or bivalent, and because of that in metaphase 1 this arrangement is there, and because of that sister chromatids do not separate. The chromosomes which are homologous and attached to each other, they separate. Okay, so you can see the entire chromosomes moving upwards, similarly moving downwards. And that is why the reduction in chromosome number. Now coming to meiosis 2, okay, so now what are we going to show? So again, now these are the cells. So now what happens is, normally meiosis 2 continues without a break. So as soon as cytokinesis 1 is over, prophase 2 will start, metaphase 2, anaphase 2, telophase 2 and so on. But in some cells there is a gap after cytokinesis 1 ends. Okay, so after cytokinesis 1, in some of the cells there is a short break which is called as interkinesis. It is like an interval. So there is a very short break in which the cell regroups itself. For example, I will tell you something. If you see over here in these two cells which are formed, now each of this cell is having only one centrosome. If you want to start meiosis 2, you will require 2 centrosomes, right? So what happens in interkinesis? An extra centrosome is formed. Normally centrosomes are formed during G2 phase, but please remember, interphase is not going to come after cytokinesis 1. Interphase is just there before meiosis 1. Between meiosis 1 and meiosis 2 there is no interphase. So how will you get that extra centrosome which you need? So in this interkinesis, all those things which are needed for meiosis 2, they are produced. If a cell doesn't undergo interkinesis, then quickly after cytokinesis 1, an extra centrosome will be formed so that you can resume with meiosis 2. So it doesn't mean that all the cells undergoing meiosis will have interkinesis. Please remember this. So this is like a break. Okay, this is like a break in between the two meiotic stages, but it doesn't happen in all the organisms. In some it might occur, in the others there might be no break. In that case, as soon as cytokinesis 1 ends, the extra centrosome is produced. Is this clear? Now let us start with meiosis 2. So what I'm going to do is, we are just going to change or alter these diagrams which we have already drawn. Okay, this will be metaphase 2, this will be anaphase 2, this will be telophase 2, this will be cytokinesis 2. Alright, so now in prophase 2, what is going to happen? In cytokinesis 1, the nuclear membrane which was formed, again it will begin to disappear. So I'm going to show a dotted nuclear membrane. Same thing. Now this is like mitosis. Then the chromosome number had reduced in each of these cells to 2. So we are going to show the chromosomes. So again chromatin will condense into chromosome and we are going to show 2 chromosomes. Correct? If in cytokinesis 1 the chromosomes have remained as they are, then these steps will be unnecessary. So then you will not have to show that nuclear membrane is formed, chromosomes are also formed, because they are already there. So depends on what happens in cytokinesis 1. Anyway, so this nuclear membrane will disappear. Then what will happen to the centrosomes? So already after cytokinesis 1, the extra centrosome has been formed and they will begin to move away from each other. Again the same thing, spindle fibers or microtubules will begin to form, and by the end of prophase 2, what is going to happen is your nuclear membrane will be completely gone. Okay, now we come to metaphase. What is going to happen in metaphase? Both these chromosomes, again the arrangement now is going to be like mitosis. Please don't make a homologous pair and club them together. That is not going to happen. That happens only in prophase 1. Now again, what do? The centrosomes have already moved towards the poles. Correct? We are studying meiosis 2 right now. The spindle fibers will go and connect to the centromeres with the help of kinetochores. So this is exactly like mitosis. Okay, what is going to happen in anaphase 2? So in anaphase 2, again the 2 centrosomes, then each spindle fiber will pull the sister chromatid. Now we are talking about sister chromatids again, because each sister chromatid will be pulled towards the respective poles. So the sister chromatids break, and again the same thing, you can see that each chromosome splits into 2. So please remember, if you are asked the question, when do sister chromatids split during meiosis, the answer has to be anaphase 2, not anaphase 1. In anaphase 1, sister chromatids do not split. Homologous chromosomes, they split up. Is this clear? Then we come to telophase 2. Telophase 2, what is going to happen? Here I have 2 chromosomes, here I have 2 chromosomes. Please remember, now these chromosomes have only single chromatid, yet they are counted as a single chromosome. Correct? So the chromosome number is going to remain the same. What is going to happen next? The nuclear membrane will be reformed. Correct? That is what I am showing over here. What is going to be next? You have to show the centrosomes again. Single centrosome, another centrosome. Correct? And last is cytokinesis 2. What is going to happen? So in cytokinesis, obviously you know the furrow will be formed, and finally you are going to have the cell which is having centrosomes over here. All right, and you are having the nuclear membrane. Okay, oh, I forgot to show that the chromosome again decondenses into chromatin, and so you will have chromatin over here, and these are your 2 cells which are formed. Each cell is having how many chromosomes? 2 chromosomes. So in meiosis 2, does the chromosome number change? No, it doesn't change. Okay, in meiosis 2, the chromosome number remains the same, but in meiosis 1, the chromosome number in the daughter cells, it becomes half compared to the chromosome number of the parent cell. Okay, is this clear? So we complete meiosis over here. So what is important is basically prophase and metaphase of meiosis 1. If you can understand that, then you practically know what is happening in meiosis. Okay, so I hope you have understood why the chromosome number reduces to half in the first stage of meiosis, and why it remains the same in meiosis 2. So that is why meiosis 2 is also called as mitosis. Okay, meiosis 1 is called as reductional division. Now, which cells undergo meiosis? So we talked about the cells which undergo mitosis, which do not undergo mitosis. Which cells undergo meiosis? So specialized cells which are called as reproductive cells, these cells they undergo meiosis. For example, gametes. Okay, gametes, for example, male gametes which are called as sperms, and the female gamete which is the ovum or the ova which is the plural. So these are formed by meiosis, and that is why if you check the chromosome number in a sperm, a human sperm will have only 23 chromosomes. That is the haploid number, half of the normal somatic cell. If you check any other cell of the human body, you will have 46 chromosomes, but sperm has 23. Similarly, ovum will have 23. Why? Because during fertilization or during the formation of zygote, the sperm and the ovum they unite. Okay, so sperm and ovum, when they unite, you will have 23 plus 23, and that will give you total 46 chromosomes. So a new organism which is formed, the first cell, it is a diploid cell. It will then continue and it will form embryo and it will form an adult. Is this clear? So why do you need meiosis? Because if meiosis was not there, then the chromosome number of a species would not have been constant. Just imagine if mitosis produces gametes, then one gamete will be 46, other will be 46. You will have a new organism having 92 chromosomes. The next one will have 184 chromosomes. So meiosis keeps the chromosome number of a species stable. Please remember, and only gametes undergo meiosis. No other cell of the body undergoes meiosis. Is this clear? All the remaining cells of the body undergo mitosis. A few of them do not undergo anything. For example, RBCs, neurons. Okay. Also remember, after meiosis is done, okay, so many of you will ask me this question that okay, after meiosis 1, 2 cells are formed. After meiosis 2, 4 cells are formed. Okay, so let me just go down. Okay, so we just did this right now. The chromosome number was 4. Then we formed 2 cells which are having chromosome number 2 each. Then we saw that total 4 cells are formed, each is having chromosome number 2. Now the question is, will these, so these are your gametes which are formed. Will these gametes undergo further division? No, they won't. They won't. So many of you think that again these cells will undergo division. No, they won't. So meiosis stops here. In case of mitosis, if you see, mitosis will continue. If there are 4 chromosomes in mitosis cell, daughter cells will have 4 each, then those daughter cells will have 4 each, and this will continue. So mitosis will not stop. But in meiosis, after meiosis 2 is complete, the cells do not further divide, gametes do not divide further. So if I ask you, in meiosis, how many times will a cell undergo interphase? Only once, that is before meiosis 1. And after meiosis 2 cytokinesis 2, there is no interphase. However, in mitosis, every time mitosis occurs, before that interphase will be there. Is this clear? So with that, we are done with this topic of meiosis. Okay, so I hope this chapter, this topic is clear. Okay, if you have any doubts, please leave a comment. If you have not subscribed, please kindly subscribe to my channel. Press the bell icon, like this video, share this video. Okay, and you can also give me suggestions on which topics do you want me to make newer videos. Okay.